Fluorescence instruments from Horiba Scientific are involved in many aspects of modern research, analysis, and quality control in the fields of chemistry, biochemistry and biology, medical research, pharmaceuticals, food science and agriculture, environmental studies, and nanotechnology. This short video will show you how Horiba scientific instruments are indispensable for your research and analysis. In the field of chemistry, our spectrofluorometers can help you determine solvent effects on reactions, temperature effects on reactions, quantum yields, dipole moments, singlet oxygen, ligand effects, cryogenic phenomena, and surface chemistry. For example, this experiment shows how diphenylhexatriene, a long thin molecule, rotates freely with a correlation time of 4.9 nanoseconds in a mineral oil. You can see how easy it is to access an additional dimension of information with lifetime measurements. Here is terbium chloride at the very low concentration of 10 to the minus 5 molar, used as a probe. Biochemical and biological applications of Horiba scientific instruments are many. For example, study transmembrane protein transfer, characterization of photoreceptors, gene expression via molecular beacons, binding curves of proteins, fluorescent corals, conformational rotamers, tryptophan phosphorescence, endogenous skin fluorescence, and pH measurements in cells. Single-stranded DNA was hybridized with complementary DNA plus a molecular beacon, fluorescing only when the beacon is open. This is an example of Foerster resonance energy transfer, or FRET. Here is an example of the fluorescence of living corals, both excitation and emission spectra, besides providing insight into symbiotic algal photosynthesis and a possible monitor of reef stability. Such spectra help distinguish between different corals. Those in the medical, cosmetics, and pharmaceutical industries will benefit from our fluorescence instruments. Examples include free versus complex drug analysis, kinetic determinations of vitamins, screening of bioassays, tooth decay, ultraviolet damage to hair, sunscreen protection factors, and photodynamic therapy. Theophylline is a vascular and bronchial medication. A polarization scan of labeled free theophylline and theophylline complex with antibody shows an obvious difference in polarization. Using known complex concentrations of theophylline, a calibration curve can be constructed. There is no interference with fluorescence polarization from other medicines, nor need one perform a separation as with radioimmunoassay. Sunscreen transmittance spectra on human skin were recorded on a fluorolog, comparing 1% oxybenzone with 1% parsol. Note the lower transmittance of parsol. This method is a quick, non-invasive diagnostic tool for accurate determination of UVA protection factors. Food science, agriculture, and environmental science can reap rewards from using our spectrofluorometers. Studies in these areas include increasing crop yields, water quality, geological transport of water, analytical determination of hazardous wastes. The U.S. Geological Survey used our Fluoromax compact spectrofluorometer to extract various components from California groundwater. Experiments like these help determine sources of dissolved organic matter in the water and the water's underground flow pattern. A comparison of Festuca chlorophyll at room temperature and cooled with liquid nitrogen, captured with our Dewar accessory, shows how the 681 nanometer peak sharpens into a doublet upon chilling. Workers in the burgeoning area of nanotechnology use Horiba scientific instruments to investigate structure of single-walled carbon nanotubes, composition of heterogeneous nanotube samples, quantum dots, nanoparticles. Here we see lead sulfide polycarbonate quantum dots fluorescence decay at 1465 nanometers over time when dispersed in trichloromethane, with dual lifetimes of 242 nanoseconds and 928 nanoseconds. A full characterization of an excitation emission matrix from a single-walled carbon nanotube sample is shown here with a helix angle plot using our nanolog spectrofluorometer plus Nanosizer software. Those in industry and security can use our fluorimeters to assist research, development, and quality control. Our customers have examined galvanizing baths, textiles, organic LEDs and flat panel displays, explosives, sol gel processes, bioactive surfaces. Using our TCSPC Fluorolog system, we show an organic LED's phosphorescence decay over the course of 15 microseconds. Horiba Scientific offers lifetime solutions from the picosecond to millisecond range. And this is a study of silica sol gel polymerization comparing polarization spectra at various polymerization times. For more information, please feel free to explore our website further. If you have any questions, please contact us or your local Horiba scientific representative for more information. Thank you.